My name's David. I'm a specialist biomedical scientist in haematology. This is our blood sciences core laboratory. This area here is our specimen reception, where we receive all of our samples from all over Tayside, Perthshire, Angus and the north of Fife. 95% of our samples are requested on a system called ICE, which produces barcodes that contain a specific ICE number for the request and the patient demographic, the test that's been requested and the patient's clinical details. Others can still come by paper and we can manually input them into our database. This blue screen here is our database, our LIMS system. So when the barcode's scanned, it loads up the tests that have been requested and the patient's demographic as well as their clinical details. The majority of the samples from all over the hospital and the porters and the drivers will come through this hatch here, and anything urgent is usually phoned ahead to this bench and comes through the pod system. That can be from Ward 15 or A&E and the labour suites as well. Once it's been checked and scanned in, it then comes across and goes into one of these racks here. This is the input side of the track. So the rack will go on the input side. It comes along and breaks a laser that then knows that there's a sample there to pick up. So the gripper will come along, pick up the sample and put it into a puck, which is one of the black circles that are circling around the track there. All the pucks have an RFID, which is married to the ICE label, which allows us to be able to find the sample anywhere in the track and deliver it for further investigation. That screen there is the GUI. It lets us know the status of all the analyzers and peripheral equipment on the lab. And this is the input-output area, so we can skip the queue here for urgent samples and stick them into that blue rack there, and it can get ahead. We can also cover the delivery lane at the end there. There's also other storage racks here, and if there's any problem samples, they'll also come out in this area. The first analyzer we'll look at is the full blood count analyzer, the Advia 2120. It can analyse 120 samples for a full blood count in an hour. It takes 170 microliters of sample to do this and separates it into different chambers in the inside of the analyzer. I'll show you in a second. It gives you a 5-part differential as well as your red blood cells, platelets and your hemoglobin. So your 5-part is your neutrophils, lymphocytes, monocytes, eosinophils and basophils. And it does this by cytochemical reactions, and then it's fed through the lamps at the top here, which is called flow cell cytometry. The information that it gets on that is then plotted onto the dot plots that you'll see here in a second. The purple shows your white cells, the blue is your platelets, and the red is your hemoglobin and red cells. The yellow also shows the flow rates of the analyzer to make sure it's working properly. We have 4 of these analyzers here, and at busy points of the day these can be running at full capacity. And we have to keep an eye on them to make sure that they're all working to full capacity. They need regular maintenance and QC monitoring as well. Up here we're coming to our auto slide, so an aliquot of blood will go onto a glass slide here, and if you see the left hand side there, that's a rack of slides. A drop of blood goes on, and then I'll show you the inside of this just now. It goes along a sort of track and it smears the blood into a triangular shape. It then stamps it with the patient's information and lab number and puts it into the carousel to be stained with eosine and methylene blue. This is a hematech for when we're making our own slides manually, and it's got a similar staining in there. This is the slide bench where we keep all our routine slides and urgent slides for microscopy. I'll talk about the microscopes when we get around to there. And here is our centrifuge. So there's centrifuges and decappers, and there's sealers as well on the track. And this automation helps us to get on with to keeping results going and phoning out urgent results to the doctors and giving us time to analyse samples as well, given the volume that they receive. This is just a manual locate system for when we have to investigate samples. We'll put them in the fridge rather than going to the large storage system, which I'll also show you in a second. So coming round here is our technical and our clinical validation. So this video was taken out of hours, so there's only one person on just now. Usually there'd be two people. One person would be on the central link, which is the technical validation. So all information that's on the ADVIAs or the ILs, again I'll show you in a second, comes over to this machine, this system, sorry, and then you can have a look to see how it's ran, if it's what it's been held for. If this, you can also tell if the machine's ran okay, what the dot plots look like. All this information here is, like the dot plots, etc., is not available to the person that's on clinical validation. So if there's anything here that's unusual, you might want to tell the person that's sitting here on the clinical validation. The clinical validation is the last point for the biomedical scientist to make sure that everything's right before sending it back out for the clinician, the doctor or the nurse to see and to act upon. So all the results will be here, and you can also review the previous patient history to see if it's normal for that patient to have abnormal results, if they're on any medication, and if there's anything, if you think something's unusual, you might want to add on further tests or investigate the sample for a clot if there's low platelets at this point. The GUI is also here if we want to just deliver samples straight away to investigate them further. And yeah, so you can see there we can just type in the sample number and it'll tell you where the sample's is on the track, and you can just click deliver and it'll come out of the rack fairly quickly. Coming up here is our IL TOPS systems. These systems monitor your coagulation. In core laboratory, the four main tests we look at is PT for the extrinsic pathway, your APTT for intrinsic, and D-dimer for looking at DIC, DVT, and your fibrinogen, body's ability to stop bleeding. It does this through endpoint analysis, so for a PT and an APTT, the samples come calcium depleted because calcium starts the coagulation cascade, so we don't want that to happen when it's getting rattled through the pod system. We don't want it to get activated beforehand. Our reagents are timed, our reactions are timed, so the reagents contain calcium. They also contain phospholipids, which act as your platelets to start the coagulation cascade. The reagents are added to an aliquot of the sample. This triggers the coagulation cascade, as I mentioned, and the time it takes for a PT for it to form a solid gel is measured by the light. So when the light gets broken, it knows that the clot's been formed, and it stops to show you that that's the time stops to show you that's the clot time for that patient. The APTTs, again, is the same principle, but it's a two-step reaction. So the first step is the aliquot of the sample gets incubated with the patient's sample, and then the calcium's added to initiate the coagulation cascade. The D-dimer is slightly different. It's immunoturbimetric, so it's looking at DNA fragments in the sample. The reagent contains silica beads, which bind to the DNA fragments, and that causes agglutination. The agglutination increases the turbidity of the sample. Again, this is measured and gives a quantification of the level of D-dimer present in the patient's sample. Going on into chemistry now, we'll come up to our storage unit here. Our storage unit can store nearly 16,000 samples, which isn't enough. We are in the process of going to be getting a new storage facility built down near at the special reception area. But moving on from here, we'll discuss an overview of the biochemistry department. So at the moment there's 3 different analyzers available in the department, but this is going to change in the near future as we're going to be undergoing a refurbishment, so these all will go into one analyzer. The 3 analyzers cover in excess of 100 different tests. The vast majority of these tests are available 24 hours a day, 365 days a year. This means that we're always able to get results out to clinicians to help them treat patients. We can test urine, CSF, bloods to help us diagnose disease and monitor the disease progression. We monitor enzymes, proteins, hormones, electrolytes, and even therapeutic drugs. Analyzers here would require to have a lot of expertise and know our analyzers inside out. They do require a lot of troubleshooting, maintenance, and have to investigate why sometimes we're not getting the correct results. Just went past the validation bench there for chemistry. It's a similar system to ours. We have to check the results and make sure that they're valid before sending them out. The storage system is also automated and knows when to throw out samples between 2 to 3 days. Down here is our microscopy area where we look at the blood cells. We're looking at white cells and the red cells and the platelets. We can check for anemia and if someone's got an infection, for investigate causes for low platelets, and we can also see if someone's maybe got a leukemia. This is actually the starting point for leukemic investigations, and is one of the most interesting parts of my job. Most rewarding. Thank you for listening to this tour. Sorry it's been so whirlwind.